Hello, everyone, and welcome. Thank you so much for taking the time to join us today and for being willing to share your voice. My name is Rachel Peterson. I'm the Chief of Staff for the All of Us Research Program. I've worked in clinical research for more than 20 years. I want to start by emphasizing how important your perspectives are. Participant voices truly shape our daily work and the future of the All of Us Research Program, and we're so grateful that you're here. This is our first participant forum that is actually open to everyone, and so I also want to warmly welcome any researchers and members of the public who are joining us to learn more. Next, I'm going to walk through today's agenda. So on the next slide, we're going to share a few research highlights. We'll spend time in discussion and question and answer session so that we can hear directly from you, and then we'll also share the winners of the Community and People's Choice Awards. A couple reminders and housekeeping items on the next slide. Science Day is a public event, so to protect your privacy, you can change your display name when you enter. Closed captions are available throughout the day. To turn on the captions, you can click the CC closed caption icon on the top right of your screen and choose your preferred language. If you run into any technical issues or need an accommodation, please email allavusscienceday@pyxispartners.co. On the next slide, I have another couple rules, and then we'll get started. As I mentioned, we want to create an open, respectful, and engaging space for everyone. So to ask a question, you can use the Q&A option at the top right of your screen, type your question and submit it. The Q&A is private, and it is monitored by all of us staff. You can also make liberal use of the chat feature as well. Reactions, share who you are if you so choose. But the questions that are submitted in the chat box won't be monitored, and it is public. So please be mindful of any personal information that you do share there. If we see questions in the chat area, we might turf them over to the Q&A side so that we can triage and monitor them. And now we'll move into our research highlights. I am so excited to welcome today 3 researchers who will have a few minutes to present their work. On the next slide, I'm going to introduce you so that you can know who they are, and then we're going to hear from them back to back as they present their work. And then we'll open up the floor for questions from you to them. So first up, we will have Annika Misra. She is a computational biologist at the Broad Institute of the Massachusetts Institute of Technology and Harvard. Her work focuses on developing and applying computational and statistical methods for personalized medicine. Recently, she has worked on polygenic risk scores for cardiovascular disease using all of us data and their clinical usefulness across different populations. Daniel Sabater Miniram is a health services researcher at the University of California, San Diego, who focuses on reducing cancer disparities in underserved populations. He uses advanced statistical and computational methods to analyze large data sets, including all of us, Medicare, and VA data, to study how demographic and social factors influence cancer risk and outcomes. And then last but not least, we have Holly Nicastro, who is a senior nutrition scientist at the National Institutes of Health, Office of Nutrition Research. She is the scientific lead for Nutrition for Precision Health, which is powered by the All of Us Research Program. She oversees the study network across the country and works to advance precision nutrition by developing tools to predict individual responses to foods and dietary patterns. So I'm going to give the floor to Annika to begin her presentation. Thank you, Annika.
Hello, thank you for the introduction. So I'm going to be talking today about polygenic risk scores and identifying how your genes influence risk for health conditions. Next slide. So many diseases are caused, at least in part, by some underlying genetic reasons, and these diseases are often thought of as being either single gene diseases or complex diseases. So single gene diseases are, of course, caused by variation in a single gene, for example, cystic fibrosis. But many cardiovascular diseases are complex diseases, and so this means that they're caused by a variety of factors, genetic and non-genetic, including other lifestyle and environmental factors and variation across not just one gene, but several different genes. These are also called polygenic diseases, and for these types of diseases, we can construct what are called polygenic risk scores. To calculate these, we compare the genomes of people with these diseases compared to those without, and we try and estimate which specific genetic variants are the most influential for causing disease. And so what these scores can do is give you an estimate of your relative risk of developing a certain disease. Your relative risk is your risk of developing disease compared to a different group of people with a different genetic makeup. Next slide. So what we've done using the All of Us data set is create a test encompassing 8 different cardiovascular conditions to start, and we've created these polygenic risk scores for all 8 of these conditions. As the field has been kind of expanding over the last decade, there have been tons of different polygenic risk scores being developed by various different groups around the world using various different data sources. And what we've done is using a model developed in our lab, kind of integrated them all into single scores so that we can kind of have a comprehensive score for each disease encompassing all the different available data. And this is largely possible due to the massive biobank that is All of Us, particularly its size and also its diversity. So this original test was created using All of Us version 7, so over 200,000 All of Us participants, and then we also performed a separate validation in our own healthcare system biobank, which is Mass General Brigham Biobank. And we showed, again, the same strong, reliable performance that we observed in All of Us. And this test covered 8 different cardiovascular conditions. So this is kind of on a spectrum of severity. Coronary artery disease is to do with kind of plaque buildup in your coronary arteries. But there's also a lot of other conditions that are risk factors for a more severe cardiovascular phenotype. So, for example, you might already be familiar with diabetes, extreme hypertension, which is high blood pressure, severe hypercholesterolemia, which is elevated levels of LDL cholesterol. And so having this test that spans the severity of the cardiovascular phenotypic spectrum is really helpful in terms of being able to identify which specific, not only cardiovascular diseases, but risk factors you might be at high risk for. Next slide. So what we found is that participants with higher genetic or polygenic risk scores had a much greater chance of developing certain heart conditions. So, for example, people in the highest genetic risk group for coronary artery disease were 3.7 times more likely to develop disease than people that were in our average risk group of for coronary artery disease. And so how we define it for this test for most conditions is that our high risk group was being in the 91st to 100th percentile, whereas our average risk group was being in the 26th to 75th percentile. So as I mentioned, relative risk, we're kind of looking at the risk of these different groups compared to our average risk group. Similarly, people in our highest genetic risk group for type 2 diabetes were 3.1 times more likely to develop diabetes than those in the average risk group. So this tool is now available as a clinical test to help support earlier prevention and care. If you want to scan that QR code, it's available to anybody in the US and also, I believe, anyone in the world. It is clinician ordered, but there are kind of a lot of different avenues for integrating this test into current clinical care. So a lot of the traditional risk calculators at use right now will take into account things like age, sex, diet, lifestyle, but many of them do not include any sort of genetic component. This is a somewhat novel addition to cardiovascular prevention methods, and it's a much more kind of integrative way of approaching cardiovascular disease prevention moving forward. So yeah, with that, thank you so much, and I will give the floor to Daniel to start talking about stomach cancer prediction. Thank you.
Thank you so much for the introduction and great talk. My name is Daniel Savitch-Minarim from UCSD Health, also part of the Center for Health Equity Education and Research, also known as CHEER. Thank you for having me today. It's a privilege to discuss our work on stomach cancer risk prediction, combining genetics, health, and social factors. Next slide. So just some background on stomach cancer. It is significant but an overlooked cancer, especially in the Hispanic and Latino communities. Today, I want to talk about what we see in the data and why it matters. Hispanic and Latino people are around 2 times more likely than non-Hispanic white people to develop stomach cancer. And there's a few hypotheses of why this is happening due to the increase of H. pylori infection, diet and lifestyle factors, and sometimes delay in diagnosis due to barriers of accessibility. But due to these factors, Hispanic and Latino people are also twice as likely to die from stomach cancer. And these are also similar in why they might be increasing, but we see that this increased barrier of getting access and this increased incidence also leads to later staging, which causes more barriers to treatment adherence and leading to poorer outcomes. And you can sort of see these on the figure on the right here, showing the 4.5 to 2, which is the Hispanic Latino individuals compared to the non-Hispanic white individuals. And one of the factors that could also be driving this is due to the lack of underrepresentation in stomach cancer research. The National Stomach Cancer Tumor Database has around 1,400 patients, and only 72 of those are Hispanic Latino. So we are missing huge genetic insights that could help improve these outcomes. So these disparities are addressable. It requires equitable screening, earlier detection, and a diverse representation in the research, which leads us to our next slide. Thanks to all the participants and all of us, we were able to get a diverse cohort of around 540 participants with stomach cancer and around 1,600 control participants without stomach cancer. Our main goal is to sort of grab all the information that all of us provides, which includes medical records, DNA information, basic information like age and background, and surveys about your life and social needs, and sort of group those all together so we can provide more targeted screening for individuals that are at higher risk for stomach cancer. Next slide. But before we dive deeper into this research, I kind of want to hear from everybody. So we'll be providing this poll. Feel free to answer. It's not a requirement. But the main question is, what do you think are the most important risk factors for stomach cancer? And as you complete this poll, you can see that some of the risk factors are biological, behavioral, and structural barriers. If you have any questions or are struggling to have access to the poll, feel free to message us too. Perfect. I think everybody's close to answering, finally answered. We can move on to the next slide. And as you can see from the poll, you're very close to what we were sort of shown looking at all of us. So now we'll look at what we actually found. After analyzing the 530 patients compared to the control group, we can see there are differences of these risk factors in the cases compared to the controls and just percentages. But as we dive deeper, I want to clarify a few things. We want to look at the odds ratios, which tells us the odds of having stomach cancer as a stomach cancer when you have these risk factors compared if you didn't. So if your odd ratio is higher than 1, that means you have higher odds of having stomach cancer. So for example, if you have stomach ulcers, you're around 7 times higher of having stomach cancer. And as you can see, one of the big pictures from taking away from this is that there are various things that can be affecting your causes of stomach cancer. And that is sort of the goal and the main takeaway is to sort of be able to have a targeted screening looking at all these risk factors. Another thing that you can look at is family history, having 3 times higher for stomach cancer, and also food insecurity, which was around 2.7 times higher risk of stomach cancer. One of the key takeaways too from this study is that we did survey questions. The survey questions were asking individuals' experience while receiving treatment. It sort of showed that Hispanic, Latino individuals and Black individuals experienced greater experience unfair treatment in healthcare, sort of showing that there's a need for cultural humility training so everybody feels welcome and respected while they're receiving treatment. Next slide. Some of the future directions that we want to take this is test a prediction tool and other data sets, and use more blood markers for genetic studies to build better risk scores that will help welcome different communities. One of the big picture goals is to include more Hispanic, Latino people and other underrepresented communities in research studies, add more social and life factors and risk tools to make it more accurate, and make sure ancestry is used in a fair and responsible way in healthcare. I want to thank UCSD, the National Alliance for Hispanic Health, and all the participants and all of us for being able to do this study. Now I would like to pass it to Dr. Nicastro.
Great, thank you, Daniel. And thank you to the organizers of Science Day for having me come and talk about what I think is such a fun and exciting area of research, which is precision nutrition. We're all here today because we're participating in this precision medicine study. But what do we mean when we say precision nutrition? Let's go to the next slide. So nutrition, or the foods we eat, can help prevent and fight conditions like high blood pressure, diabetes, stroke, cancer, and more. But one size fits all does not work for nutrition. We're all starting from a different place, different environments, different cultures, different biologies. Our bodies deal with food differently. So precision nutrition means using information about individuals to tailor our dietary advice. Some past precision nutrition studies found that information about people can be used to predict whether specific foods will make your bodies respond in a good way or a bad way. And different people responded completely differently to different foods. Some of the reasons for this we know comes from the bacteria that live in our intestines, but we're still learning other factors. But these past precision nutrition studies have been somewhat small, and they didn't necessarily study diverse groups of people. So we launched Nutrition for Precision Health, powered by the All of Us Research Program, or as I'll refer to it, NPH. In NPH, we're looking to study a large and diverse group of All of Us participants and comprehensively study why people respond differently to foods, with our ultimate goal being to be able to provide individualized dietary advice so that we can all optimize our health. Next slide, please. So by the end of NPH, which is ongoing now and will wrap up in about a year, we are going to unlock personalized nutrition insights by learning how different people respond to different foods. We're also going to strengthen the All of Us Researcher Workbench by adding new rich data types related to nutrition, physiology, different omics, and lots of other data that's not currently there in the workbench to enable future discoveries. And we're also going to test new and better ways to measure how we study what people are eating. Next, please. This is what it looks like if you become a participant in NPH. We'll start with a screening, typically a phone call, where we determine if you're eligible. We have very broad eligibility, and we want to welcome everybody. The biggest limitation, I think, for most people is geography. The study is only conducted in 6 states because we need to bring you into the study center. It's a little different than being able to participate fully online. But if you're eligible, you can give your consent. This is a separate consent process from All of Us. And then you'll come in for your first study visit. There, you'll answer a few questions and talk about your diet. You're also going to be sent home with a lot of instructions and materials and different wearable technologies. For example, we'll give you a continuous glucose monitor. You'll also be asked to wear something on your wrist that's a lot like a watch that tracks your activity and your sleep. And over the next 8 to 10 days, you're going to go about your life as you normally would, but you're also going to be collecting data. You'll answer some questionnaires remotely. You'll collect information or take pictures of all the food that you're eating. And you're going to continue to use this wearable technology to collect different things. And at the end of that 8 to 10 day period, you'll come in for your second study visit. The second study visit takes about 5 to 8 hours, and that's because you're going to come in and you're going to consume a meal, and we're going to track changes in your body over time in response to that meal. This study participation also has a lot of return of value for participants. You're compensated for your time. You'll receive educational content to learn more about nutrition and diet. You'll also receive individual results, things like how your own blood glucose is changing throughout the day. You'll get insights on your typical diet, on your activity and your sleep, and you'll get your lab results and more. And at the end of the study, you'll receive aggregate study results, so what we find globally overall from this study. Next slide, please. So we really hope to be able to benefit general knowledge about nutrition and our health. We're looking to discover how what we eat influences our risk of diseases like heart disease, diabetes, obesity, and cancer. And very importantly, and what really hasn't been done before, we're looking to learn why certain ways of eating work better for some people and not others, so that we can then give you individualized advice. If you're interested in participating, you can check out nutritionforprecisionhealth.org or the QR code. I hope that today I've sparked your interest either as a potential participant or just as a community member who's anxiously awaiting our initial results and findings of the study, or the data that's going to come from this study for continued discovery, both of which are going to start to be available in the next year or so. So I want to thank you for your time today. I want to thank our participants, and I'm going to hand it back to Rachel. Thanks.
Thank you so much, Holly and Daniel and Annika, as our researchers, for sharing your work with us today. Now we're going to move on to the question and answer phase of our session. Before we begin, I'm going to have two of our program leadership join us on screen as well. So they will share a couple updates about what you can expect from the program. We're joined by Dr. Minky Lee. Minky is the Acting Chief Engagement Officer for the All of Us Research Program. Since 2017, she has worked alongside All of Us awardees in the community to identify and disseminate best practices in the engagement and retention of participants as well as researchers. We're also joined by Dr. Josh Denny, who is the All of Us Chief Executive Officer, the CEO, and he's been involved in the program since before its inception. He helped to develop the program's initial scientific blueprint as a member of the National Institutes of Health Precision Medicine Initiative Working Group. He's also a researcher who uses the data in the workbench and a physician. Josh, would you like to say a few words before we start with the questions from the audience?
Certainly, Rachel. Thank you so much for the opportunity to say a few words, and it's really great to interact with you all again. This is our first time we've had a participant forum that's truly open, and I know many of you may have joined this as participants from the CEO's email or other ways you saw this event, and I'm thrilled that you joined us today. I have to say, as we near the close, I want to take this opportunity briefly to say thank you to everyone who made it possible, especially on the All of Us team, Sydney McMaster, Jessica Lee, and Minky, who's on here as well, and say a few words in a minute, the Pyxis and technical teams, and all the people who submitted abstracts in a competitive way are great speakers as well. And what really stood out to me was just the not just the breadth and depth of the science, which has progressed so tremendously since we started this program, the hope and promise that we hope to see, I feel like we are actually seeing with the matriculation of the science here that is going to have real impact and be relevant to everyone. And you saw that highlighted with some of the talks directly of how we are seeing things that we couldn't see with studies before, because we have the breadth of the United States covered and the diversity of genetic ancestries that have never been covered before, the environmental exposures, the medication exposures, all those things that really make a difference to make discovery possible. And so the participants out there, I want to say a huge thank you for being with us, for making this possible. And it's what you do and what you contribute that make all of us possible and make all of us so special. And to the researchers, I also want to highlight there's a great deal to look forward to. We're going to have what will be the world's largest and most genetically diverse data set ever released that's linked to health outcomes, which is so critical to uncovering real health change for people. That's going to be coming in the next couple of months. We'll also have things like long-read sequencing and methylation data. We'll have our first proteomics data set to understand proteins and their impact. We will have an opportunity for researchers to propose ideas for what is great science and great ideas using what's called an XL1 mechanism to do new research on our data set, which becomes available to everyone. And for participants, if you haven't logged in recently, I'll just ask you to take a look to your portal, sign in. If you haven't done it in a while, you'll have to reset your password probably. Hopefully through today, you've seen the power of all those surveys. And this is just a sampling of the science that's happening and the studies that are coming out literally every day. I get an email in my inbox with 1 to 3 studies that happened in all of us. Take a look at the publication list and look at the summaries of what's happening out there. We are going to have what's called our All of Us check-in coming soon. That is our first time we've asked you the same questions that we asked you when you enrolled. We're going to ask a lot of those again and some new ones on things like oral health and more stuff, items on women's health and some items like that we haven't addressed well before. And so that's going to be coming up for most of you around the summer timeframe. And to conclude, one quote that came from a participant at one of the sessions today, I think sums it up best as I think about what we're trying to build here and what we're doing together as researchers, staff, participants. This was from a participant today. The data I contribute today will outlive me, eventually shared by a researcher not yet born, using technology not yet imagined, at a conference not yet planned. So that's the vision of all of us. We are building a foundation that will last for decades, and I'm just thankful that we get a chance to celebrate that, to do that here. I'm really looking forward to having your questions in this time of discussion. So Rachel, I'll turn it back over to you.
Thank you so much, Josh. So just a quick reminder to everyone on how to submit questions. You can use the Q&A option at the top right of your screen. If you're bold and you want to ask a live question, we are recording this. Go to the chat option on the top right and you raise your hand, and then I'll be calling on people throughout. We're going to do our best to answer as many questions as possible. I think we're going until about 5:20 with the Q&A. So first question that we have from the audience, I'm going to turf this question to Annika and then to Josh. How well do the polygenic risk scores that you presented on, Annika, perform across all ancestries?
Yeah, that's a great question. So polygenic risk scores, of course, are kind of optimized for the populations that they were developed in. Many of these have been Eurocentric for building on data from kind of the past decades. So many of the scores in general that we kind of use to build our panel are exhibiting stronger performance in European populations. Off the top of my head, for example, our coronary artery disease score in the all group, which is predominantly European, has an odds ratio of about 3.7. So you have a 3.7% or risk compared to the average risk group, whereas those that are genetically similar to people with African ancestry have an odds ratio in the high risk group of about 2.5 compared to the average risk group. So, as we kind of incorporate more methods and more GWASs are kind of continuing to come out, we hope that this will kind of attenuate over time and match more similar performances, but we feel that the performance of these scores is still strong enough within each group that it's still helpful enough regardless of what your ancestry is. Another example is for severe hypercholesterolemia, which is elevated LDL-C levels. In the European group, it's about 4.1 compared to the average risk group and the high risk group, whereas in those that are similar to people with African ancestry, the odds ratio is actually higher. It's 4.6 compared to those in the average risk group. So there's a lot of different reasons and moving pieces of these different scores. And if you want a better picture of kind of the more tabulated data, then the paper is in press and will be out in a few weeks, and we break it down all there as well if you want more granularity.
Thank you, Annika. I think you really dove into the detail of that well, and it's just a tremendous amount of work.
Yeah.
I'll just stepping back a bit on this. One of the things that's really important about all of us is there are all this excitement around the way to use polygenic risk scores to calculate risk of conditions. And I, as a practicing physician, I would, have patients in front of me. I would calculate risk scores based on the factors I knew about, and it's at the high level: good cholesterol, bad cholesterol, blood pressure, age, do you smoke, do you have diabetes, are you on an antihypertensive medication, things like that, and that's it. And these polygenic risk scores provide, she was talking 3 to 4-fold the risk if you're in that highest risk group, on top of all those kinds of scores. They give us something that we don't measure and we don't practice with as, clinicians seeing patients. And importantly, those have all been derived generally from people of European genetic ancestry, people who self-identify as white. This is the first time we have polygenic risk scores that have data behind them that are, look like the United States. And we have, as your great examples, that the data are performing better and working across different ancestries. It may vary some, and that makes sense, but it largely works.
Yeah.
Thanks, Josh and Annika. Kind of building on that, Josh, if I'm a participant who provided a blood sample, how are the samples, already being used to generate data? And then how does that help to lead to like what Annika's work generated? Can you kind of talk about that?
Yeah, the first thing we did with the data that you contributed as participants was to extract DNA and generate genetic data. So these whole genome sequences, the over 3 billion base pairs, the A, C, T's, and G's that define our genes and such. And we've made that available to researchers. And from that, a lot of the studies you saw today have been done. And then the other samples, just with this next data set, we will have the first releases of other kinds of data generated from those. So it includes the proteins in the blood at scale, proteomics we call it. It's going to include some measures of what we call the exposome. So a way using for researchers untargeted mass spec that is calling a number of different exposures from the environment that will help us understand the risk of some of the chemicals in the environment, as well as other metabolites that we might have in our bodies towards disease, to predict risk of having type 2 diabetes, for instance, is one of the questions that's being asked. And then we'll continue to generate other data from that. And so those data will be coming out more over time. And one of the real powers is we have those samples before, for a lot of people, disease occurs. So those will help us predict, we think, disease risk over time.
Thanks. Speaking of disease risk over time, Annika, is anyone looking at these sort of like polygenic protection scores? So not like a polygenic risk score, but a polyprotection score or genetic variants that would confer resilience or resistance to coronary or heart disease? And could that be important for sort of looking at novel or new drug targets?
Yeah, for sure. I think there's been, I don't know about the notion of a score necessarily, but there's definitely been a large amount of research done on that, and it's still ongoing. A famous example is probably a PCSK9 loss of function variant, which kind of led to the development of PCSK9 inhibitors, which kind of dramatically lowers your LDL, your cholesterol levels, and your risk for coronary heart disease. There was also another paper that came out maybe just a few weeks ago in the Journal of American College of Cardiology that looked at almost exactly this: people that had kind of a high risk for CAD, but actually low observed plaque, and looking at which variants they might have had that were protective. So I think it's still early days. There's ongoing research. I don't know about a score, but that's definitely a good idea to look into in the future.
Thanks. This question is for Daniel. Were there any risk factors on your table on slide 14? Were they co-adjusted or adjusted for anything else?
Yeah, great question. They were sort of done univariably, but they were also adjusted with simple demographic data like age and race. but then they were all ran independently. We
Okay.
have a multivariable that we'll be releasing, showing them all together, but the results were very similar.
So then looking at the odds ratio table that you had, what should I do if I have a family history of gastric cancer and I've had gastritis, for example?
Yeah, that's a great question. I think what you can do is just be proactive and maybe talk to your primary care provider. They're the experts and they'll be able to sort of lead you on this journey of cancer prevention. So just bringing it up to them, making sure that's addressed and noted in your EHR data or your electronic health record will help you lead into sort of like if you need to be screened earlier or when you need to be screened for gastric cancer.
Thank you. This question is for Holly. How do I get involved in Nutrition for Precision Health if I'm in one of the states that's enrolling? And then on the flip side of that, how could participants in states where there are no sites support the study or just learn more about precision nutrition?
Yeah, thank you for that question. If you're in one of those states, check out the link we provided. It's nutritionforprecisionhealth.org, and it will walk you through next steps and give you phone numbers where you can directly reach out. You can also go into your participant portal and you should see an option to learn more or not about NPH. Now, if you're not in one of those 6 states, and I'm in the same boat as you, I would love to participate, but I'm in Maryland and we don't conduct this in Maryland. Please stay tuned for future opportunities. One thing that's really exciting about our study is we're embedding some of these nutrition tools within the All of Us data collection ecosystem. So that will equip us to be able to do future studies for nutrition.
Thanks. Staying on the theme of nutrition for precision health, if people do respond differently to foods, do you think that the microbiome plays a role, and are you collecting stool samples to assess that?
I do think we're going to find a role for the microbiome based on some past studies and early hypotheses. We are collecting stool. We're collecting stool from all 8,000 participants, and we're going to repeat stool samples on several participants who undergo later, deeper dives into some controlled feeding studies where they follow different diets for different days. So we're going to be able to look at the composition of and the diversity of the gut bacteria, as well as some metagenomics and metatranscriptomics.
Thanks. One participant says that they participated in the Nutrition for Precision Health study in 2025, and it was really well run. When do you think results will be ready to share?
Well, thank you for your participation. Can't do this without you. We are still ongoing, and we expect to keep enrolling participants through early 2027. So we want to have 8,000 people come through, and it's pretty intense when you are involved. So it does take some time. We will wrap up enrollment early next year in early 2027. And our big analyses can really start once that whole data set is in place. So I think you're going to see some early results start to trickle out later in 2027 and into 2028.
Thank you. This question is for Minky. I joined All of Us in 2018. Is there a plan to get updated blood or lab samples from participants at some point, or is everything that's being generated based on the original set of samples?
So currently it's being based on the original set of samples. I think the hope is to collect updated samples at some point, and we are collecting updated data through surveys coming soon, like Josh mentioned, the check-in survey. So please check the portal for that later this year. It will depend on capacity of the program and funding and all of that. So we will keep an eye on that. But there are opportunities like NPH and other partnered research studies where you might have an opportunity to donate and contribute other samples. So that's also something to stay tuned to.
Thank you, Minky. Now, Josh, I heard you and Holly both talk about omics. So whether it's, metabolomics or proteomics or other omics, can you just kind of re-review what omics is? And there are some more complex questions that we have from participants around how are the proteomics and metabolomics data getting collected? And is there a platform that you're using? Are these data available? Will they be available? And so forth.
Holly, you want to go first? You have more omics than I do.
Sure. So when I say that we're studying the microbiome, for example, by the ohm or the omics, we're looking at the totality. We're looking at the big collection of everything that's available in your gut. Same thing, we're also looking at metabolomics. That means we're going to take a blood sample and look at all of the different metabolites. The term, it does feel kind of newer as the technologies emerge and allow us to look at all metabolites that can be measured in a sample versus just glucose or just insulin.
So, and so, one of the principles of precision medicine is really the idea that we're going to be moving towards more data-driven medicine at the scale that's much deeper than what we can keep in our brains as we see a patient, or what we would just measure with the typical blood test that you see in your electronic health record, your patient portal. And so it's really moving to scale of that comprehensive protein assessment, the comprehensive genomic assessments of all the ACTs and Gs you have in your DNA. We look at the RNA that your DNA, is made from your DNA. We look at the proteins and that test. So getting to one of those questions sounds like a deeper level. So right now, that first 10,000 set roughly that will come out is based on the Olink platform. It's one of the platforms that looks at proteins. And I believe, I can't remember if it's the 5,000 or 7,000 protein spec that is used for that. But that's generated Olink. We are actually looking for hopefully generating another large scale proteomic assessment that will go much larger than that 10,000 set. We'll have the exposome data is based on compounds we find in your blood, and it's tuned to those that are more exogenous, but meaning outside the environment, outside of what your body produces, but it'll also get some of the metabolites that your body is producing as well in that. And so that's a bit of a metabolome and a exposome if you put those two together. The timelines will be this summer for the proteome, for the transcriptome, and some of the others will be a little bit later.
Thank you. Minky, we heard from more than 40 researchers today, and we had several thousands that were registered. Can you talk a little bit more about, can anyone access the data? How does researcher data access work?
So our data platform, when you go to researchallofus.org, it's running on a passport model, which means there's tiered access at different levels. Anyone here can go right now and look at the public tier. And we have a lot of information already available there. You can look at data snapshots. You can go to research projects directory, which I love sharing with participants, and you can see if any of the research topics you're interested in are being researched right now, because we require all of our researchers who have access to our data set to provide a description of what they're working on, and we make that all public. So there's a lot of transparency on what types of projects are being conducted with our data set. If, as a researcher, you want to gain more access at the individual level, and I want to caveat that with all of our participant data is de-identified before it's provided to researchers in the data platform. At the public tier, the aggregated level is shared, but if you want more detailed individual level data, you do need to register as a researcher, and also we require researchers to agree to a responsible a data use agreement and then complete a responsible conduct of research training, and we take that very seriously, and we have a team who monitor whether researchers are following that guidance that we provide. Did that answer that question, or was there more?
No, thank you so much, Minky. So Josh, building on that, the electronic health record that participants can choose to share, how is that used in research? How is it gathered? And then what do researchers see on their end?
The electronic health record data is really critical to the research we do. Almost all the studies that you see end up using electronic health record data to help define disease conditions. And importantly, when we link in electronic health record data, it has a record of time of it. And some of you have probably been with the same medical center for decades. We see this. Some of the medical centers can share decades of information, which is really important. We're actually in my research lab doing a study where we're looking at people who have high blood pressure in kind of like their 40s, 50s, and 60s, and the risk of that towards whether or not they get dementia later in life. And so that's going to happen after 65 and 70. And so to do that, you have to have a really long-term period. And what we're finding actually is there's certain, it looks like, and we're still working on it, so I'm not going to get into specific drugs yet, but it looks like certain of those drugs, provide more protection to developing dementia than others. And so that could be a really important finding for a vexing condition with Alzheimer's and other dementias. We're also other people are looking at kidney disease and protection of those. So that longitudinality with the electronic health record really helps you do that kind of work that you couldn't otherwise do. What the data look like is it is, as Minky said, it's all de-identified. So that means we don't have any of the obvious identifiers. It's taken from all sorts of, there's all these different health record systems out there. You may know some names: Epic, Cerner, Allscripts, Athena. There's so many systems out there. We actually put them all into one data model. So we harmonize the data into one kind of common data model. It gets structured and then made available with the identified data to researchers. So they just see those data in databases that they can query in one way across all these different resources and all these different EHR systems and all these different sites that provide the data. And literally, we've actually put data together from over 1,000 different places. And we keep working on expanding that to keep closing those gaps, because most of you, 95% of you, have said you can go get that data and put it together, and we're still working on gathering it. We'd love to actually reflect more of it back over time and let you know also what's in your electronic health record. We did that for a little while with our prior portal, and we might be able to do something like that more in the future as well.
Thanks, Josh. We have 3 minutes of remaining Q&A, so I'll be fast. Holly, when we think about a partnered research study, what's the difference really between All of Us and Nutrition for Precision Health? How are they same and how are they different? And then Josh, are there other partnered research studies that participants could take part in right now or in the future?
So we refer to All of Us as the parent study. So Nutrition for Precision Health is embedded in All of Us. So all of the NPH participants start as All of Us participants and then enroll in NPH. And this has a lot of benefits for our study. This means even when the Nutrition for Precision Health smaller study ends, that data is going to live on. The researcher workbench will be there. The electronic health records will be there, and they'll continue to update. Our nutrition data collection will have ended, but we'll continue to be able to follow participants to see, okay, who is or is not getting heart disease based on their diet back in these days. So that's the way we think of a partnered research study. All of Us is the big picture, and we can conduct partnered research studies within.
And right now we have a study called Eyes on Health, which is getting eye imaging data. It's doing plain pictures of the back of the eye, and it's also doing a more detailed picture. It's kind of, you could imagine it like maybe a CT scan of the eye, except it doesn't use radiation, it just uses the light. And that really clear, detailed picture, we think, will help us predict things like neurologic disease and cardiovascular disease, maybe other health conditions as well. And that's being done at a handful of sites also. So those sites that recruit people to bring people in occur from different sites. And then we release other things. Holly talked earlier about some of the Nutrition for Precision Health tools being released to everyone. And so we do things like that. And then we hope to do that in the next couple of years, where we'll be releasing more of those kinds of tools and more of those items that can come through the portal as well.
Thank you, Josh and Holly. So we'll do our last question focus on tools, future, technology. Josh, there were a lot of interesting AI-related talks today. Speakers are likely familiar with how agentic coding tools like Claude Code are speeding up research outside of what are commonly called trusted research environments. Are there currently, or is there guidance on the responsible use of these tools with data inside the researcher workbench while protecting participant privacy?
So we have general guidance for all researchers on how the data are to be used, which includes them being used within our cloud. And so those data aren't to be shared into AI systems and large language models. And so, you know, those rules were written a long time ago. They apply to now, and they protect the data now as well. And obviously, as we need to update things, we always look at that, and we have some advice specifically around large language models and how they can be used. Right now, a lot of people are actually using LLM to help them write code to query the database. And my team routinely uses systems like Claude Code to help us you know, generate and refine what the statistical code should be and things like that. And so there are some examples out there of ways to do that well. But the all of us data, like the participant level data, isn't going to those environments. It is people, you know, helping debug their code in order to with these tools. Now we're actually working on systems where we can put that side by side, so that, you know, researchers coming in can use a large language model in a protected way to help them write those analyses real time, faster, and better in the process in a way that's safe and protects patient data as well. So I think AI is tremendous in terms of what it's going to help us. We have to make sure to use it in a way that's safe and protects participant data. And I think we can do that.
Thank you so much. Thank you, Josh, Minky, Holly, Annika, Daniel, for answering our questions. And we're going to go to the next slide, please. Just how everybody was asking questions. I have a question to ask of you. If you are participants, please want to let you all know, if you haven't already this year logged into your individual participant portal account, you can access it at joinallofus.org. Enter your username and date of birth for extra protection and security, of course. And then if you haven't already, you may be asked to reset your password. We have a new sort of portal experience online. And the reason that this is important is because many of you, about 600,000 of our participants later this year, will become eligible to complete our reassessment, which is the All of Us check-in, which will have a series of both repeated questions that you may have answered 2 or more years ago, as well as some really interesting scientific new questions. And so when we're ready, we'll reach out to you individually. You'll get an ask to do that. But if you go ahead and take this step now, it'll be a lot easier when the time comes to complete this activity. So thank you so much, again, to our researchers for sharing your work with us today. And we're going to transition to a quick poll and then stay on the line because we're going to announce the Community and People's Choice Awards. And we want to hear from you. So let's see. Here is the poll on your screen. You can take a minute to complete it. Your feedback is very valuable. It helps us to shape how we as the program continue to communicate and engage with participants like you. The 3 poll questions are: how satisfied are you with today's participant forum, this 4:30 to 5:30 session? Did today's participant forum help you better understand the purpose of the All of Us research program? And do you agree? Does the All of Us openly share information in a straightforward, plain language way? So we'll give you a moment to respond. And just a reminder, your answers will not be shared individually and the poll results will not appear on the screen. But thank you so much. All right, so we will go ahead and review them and as we look to continue to improve the program. And as we begin to wrap up on the next slide, we would like to take a few moments to recognize the outstanding contributions from today's presenters. First, we're going to present the Community Award. This award was selected by our abstract reviewers prior to the event, and it recognizes the researcher whose work most clearly highlights the potential of their research and reflects the importance of communicating science in a way that connects to people and their communities. And then we'll have the People's Choice Awards, which was voted on by all of you, our event attendees, and recognizes the 5 presentations that resonated most with you. So I am so pleased to announce our community award winner is Dr. Adriana Visbal from the University of Houston downtown with her abstract Computational Reclassification of BRCA 1 and 2, breast cancer 1 and 2, variants of uncertain significance in America's ancestry populations using the NIH All of Us research program. This award was determined solely by the abstract merit itself, but it is fitting that she receives our community award because beyond her outstanding research, she actively organizes and hosts local community cafes that educate and empower individuals about BRCA genes, breast cancer genes, and health, demonstrating her commitment to translating science into community impact. So congratulations, Adriana. And then our People's Choice Awards, we have Melis Sahinos from Vanderbilt, Dr. Mole from the Queensland University. We've got Nick, Krista, and Jonathan as well. So congratulations to all of them. The breadth of their topics ranges from wearable devices and cardiovascular risk, different hidden genetic links between heart, kidney, and diabetic health, characterizing Alzheimer's disease and dementias to support healthy aging research, the association of disability status with engagement in physical activity, and then looking at behaviors and sedentary behavior during the pandemic. So totally vastly different topics that won the hearts of our attendees today. So congratulations to all of our winners and thank you for sharing your work with us today. And I want to just thank everybody for making today's event such a success, your attendance and participation, as well as to all of our planners. And I want to specifically invite Dr. Minky Lee to share her closing remarks with us as she oversaw this whole event.
Thank you, Rachel. Thank you, everyone, again, for joining us today and for your engagement throughout the whole day. I think everyone will agree it was a packed but amazing day to be here with you today. As the program's very first event where participants and researchers were brought together at such a large scale, we had hundreds of attendees attending different sessions throughout the day. I think it was a great reminder that participants and researchers are not always two completely distinct groups. There's a lot of overlap, and research really needs to move at the speed of trust with communities, research participants, and researchers working together in true partnership at all levels of the research process. We hope that this event and everything we discussed in all the different sessions help us to remember that and help us all of us move together to improve health research outcomes. If you missed any part of today's sessions, all recordings will soon become available and remain on the same exact platform for the next 90 days. So please log in and feel free to review those again. I know that some of the earlier sessions are already available, so kudos to the team to making that happen already. Registration to join the platform and view those same recorded sessions will also be possible for the next 90 days. So we'd love for you to share the link to the Science Day website with anyone you know that couldn't make it today. We truly appreciate your time and contributions. Thank you for making the All of Us research program possible so that we can have events like Science Day again. And we hope you join us again next year. Thank you, everyone.